welcome. Welcome in this online Journée Louise Harel Day, organized by the Société Française du Cancer. I'm pleased to welcome you. So it's rainy, at least in Paris. So just to say that, of course, everything was planned 9 months ago. This is why we're doing this online. It was just because it was rainy on this day. Everything was planned. It was not all organized in just 1 month. No, thank you to be connected because it's a bit difficult on these days. Thank you for the organization. I will say it once again at the end of my talk. So I would just like to introduce in few minutes the Société Française du Cancer, because I think that some of you may not know this French Society of Cancer that is now more than 100 years. And to say it briefly, the goal of the Société Française du Cancer is to have researchers in the cancer field, of course, and clinicians discussing and progressing in their knowledge about cancer. And so we are organizing seminars like this Journée Louise Harel that is every year, except last year, of course. We're also supporting seminars, and we are organizing webinars and supporting all the actions that may help expanding the knowledge about cancer. Just to present you in a few words, the Journée Louise Harel for this year we will discuss, and it will be presented by Daniel Louvard and Annick BELLAN in a few minutes. 2 years ago, it was about dark genome and cancer. 3 years ago, it was about evolution and cancer. Here also in Astucurie, in Edinburgh. So you can see the field we are embracing in this Journée Louise Harel. We are also organizing every month a webinar. It was sometimes on research side, sometimes in physician side. For instance, we have been discussing about neurosurgery, how we can do the surgery while the patients are awake. We have been discussing about also artificial intelligence and in cancer, about clinical trials. How do we run a clinical trial? That can be something interesting for the scientist. We're also organizing some courses on two-day courses. For instance, last year it was about DNA repair in cancer. We are also supporting a lot of meetings. So if you're organizing a meeting for research, for instance, and you lack funds to organize this meeting, you can contact Sophie Chervet, our director of the SFC, to ask her how can I apply for support to organize meetings. And so we are supporting meetings for physicians as well as for researchers. For instance, we are supporting meetings about stem cells in cancer, about DNA repair in cancer, about metabolism in cancer, genome instability, and such questions. So don't hesitate. We have a lot of other smaller actions, so don't hesitate also to check on the website to see what we're doing. Last but not least, we are publishing since the start of the 20th century a journal called the Bulletin du Cancer, that is the only journal where you can publish in French papers, and is indexed in PubMed, so it can be helpful, especially for the youngest to publish their first paper, but you can also publish in English. You don't have a big impact factor, as you can guess, with a French journal, but you can at least reach French-spoken people, so don't hesitate also to check the Bulletin du Cancer website. To finish, I would like to thank very much Sophie Chervet, our working people in the SFC, that is our director now, and who has been organizing this meeting for the last 9 months, more or less, and especially she did a tremendous work on these last weeks to organize all this online. It has been really a lot of work, and clearly I think it's doing very well. So thank you very much, Sophie. Thank you also Daniel Louvard for accepting this invitation and for organizing this program, and so contacting all these important people in this field. I'm very happy to see this presentation. Thank you, Annick, also to organize every year this Journée Louise Harel. And I will let now speak maybe Danielle to introduce this day and to introduce the presentation and this field. Thank you very much.
Well, first of all, glad to see many people attending, even online. We all of us would have much preferred to be in Paris today, sitting in the amphitheater. But that's how is life. To tell you a little bit about the context and the content of this day dedicated to the memory of Louise Harel. It is by the end of 2019, I was contacted by my colleague and friend Annick BELLAN, who asked me if I would be willing to organize a program for this Journée Louise Harel in 1920. I gladly accepted. Well, you know the story. I contacted several colleagues who are our invited speakers today, who were resilient enough to actually follow the progress of the pandemic and follow my final suggestion a couple of weeks ago that there was no way to hold the meeting in Paris at the Curie Institute, but the best will be to maintain, because we could not postpone once again this meeting, and then it will be done on live, which is where we are today. So when Annick asked me to to put together a program, we discussed about several possibilities. And I said, you see, there is one aspect which actually triggered my attention and interest for years already. Well, we know that cancer research, cancer biology has made fantastic progress in understanding the origin of cancer, the genetic, indeed, basis of the rising of tumors, and very powerful technology have actually followed and helped to decipher the nature of the mutation that actually encompassed many parts of the genome. And based on that, we understood better some aspect of the integrated network of and change of signaling pathway due to the mutation of key gene, including oncogene and tumor suppressor gene. Well, that's very helpful, and indeed these progresses continued. We have now the possibility to do single cell analysis, to do much better imaging than it was 20 years ago or even 10 years ago. And I think with this tool, that people have already discovered or established firmly the origin of the cell heterogeneity and the great diversity of cells that actually are present in tumors, either the primary site or the metastatic sites. So, and this, for me as a cell biologist, it was another demonstration of something that we know very well for normal cells, as well, of course, as the gain of function that were acquired by tumor cells, which is depicted in two words: cell plasticity. What is cell plasticity? In fact, cell plasticity is a well-known process. It represents the dynamic evolution of living organisms. This dynamic evolution has, of course, led to genetic changes, mutation, and also epigenetic changes, which, of course, are now in the front line of the research in cancer, but not only in cancer, but also for other chronic diseases. So this issue leads to the question, what is the base of cell heterogeneity? How does it work? What are the mechanisms of that? And what are the consequences of that for understanding a disease like cancer? And these drive actually several issues, such as what is a cell of origin? What is the nature of the cell of origin of tumors? Whether we deal with hematopoietic tumors or whether we deal with solid tumors, this is a very important question that was addressed years ago, decades ago, almost a century ago, but was not amenable due to the lack of technology allowing to carry on the experiment and provide proof and demonstration of what is the cell of origin. The consequence of this observation leads to the principle, what are the mechanisms of resistance during treatment of tumors and what are their intrinsic basis for the resistance of cells. And as you imagine, once we understand that, and we understand that better and better, this leads to the question of, if we understand better the resistance, we can think about new therapies, which is, of course, a very active part of the research on cancer biology and clinical research as well. And this, in my opinion, these two questions, what is the origin of this, what are the origin of tumors and how do they evolve, and what are the mechanisms of resistance? And we know today that many cancers can be efficiently treated, at least for some localization. In other situations, of course, the success has been not so great, and we are still struggling, and the clinicians are still struggling with its aspects. And the concept, what we need actually to understand, once we will be able to treat a secondary site where the tumor has moved and to deal with metastasis, a major step in cancer research and cancer treatment will have been achieved. The program today was designed with in contacting different people that have made outstanding contribution in their field, in this field of cellular plasticity, and we and not only in developing new concepts, new technology, and also to address the question of cellular plasticity, microenvironment, which play an essential role in the tumor development, and that leads to immune response or the lack of immune response, which are based the immune therapy of cancer today. So I think I'm very glad to thank first la Société Française du Cancer to actually organize this meeting based on this topic. Second, to Annick BELLAN, who convinced me that I should actually try to play a role in building this day in the memory of Louise Harel. And third, to all participants who are here today and 6 speakers that were invited to participate and contribute and deliver the best of their current research, and also to let us know their thought and how they see the future. Having said that, I think I will leave the stage to Annick to tell us about the Louise Harel. And again, I wish to all of you a great day. I'm sure we are going to learn a lot and make new friends virtually, but hopefully we will not stay as virtual entity, and soon we will be able all to shake our hands and give a great hug to all of our friends when this pandemic will fade away, which I hope will come soon. Thank you.
For organizing this meeting, very, very much. Thank you very much. It's a great meeting, and I'm sure all others will enjoy this day. Before we go deep into science, I would like maybe to introduce Louise Harel. You may wonder why this day is called Louise Harel. Well, it's actually because, if I can have the first slide. No first slide. Yes. Thank you. Okay, so Louise Harel was born in 1925, and she died 5 years ago. Why this meeting is called Louise Harel? It's because she originated this meeting, actually, because she has created in the early 80s the Growth Factor Club, which is the ancestor of the current Journée Louise Harel. Who was Louise Harel? She was born, as I said, in 1925 in Algiers, which is in North Africa, but was actually France at this time. And she was actually very bright because she's got her first diploma very early, as she won the Grand Concours de Bébé, which is the game to select the most beautiful baby, and she was selected as the most beautiful baby in Algiers. And you can check that she was really beautiful. Now, her second diploma, she got it in Algiers as well. It was the baccalaureate. And she's got it in a year under conditions which were very bad, because actually all the Jews were ejected from all the high schools and all the schools. It was in 1942. And she stayed in her high school thanks to her father's medal during the First World War. He had the war medal. He had very much courage during the war. And thanks to that, she was able to stay in the school and to get her baccalaureate. Now, her third diploma was a PhD in chemistry at the Sorbonne University, so she was a chemist. But she decided to switch to biology and joined the Cancer Research Institute in Villejuif, where she met her husband. And she started to make very important discoveries in biology. In 1971, she made the first observation of non-coding RNAs in mammalian cells. The paper was published in Nature New Biology, and the title was Homology of Double-Stranded RNA from Rat Livers Cells with Cellular Genome. And we all know now the importance of these double-stranded RNAs. Then she discovered that there were kinases associated with chromatin in 1976, and we all know the importance of these kinases in the cell cycle. Finally, in the early 80s, she switched from chromatin to signal transduction because she was really interested in discovering what makes the difference between a normal cell and a cancer cell. And she published several papers. For example, the effect of EGF on the ATP turnover induced by growth factors. And we know now the importance of this signaling pathway. And then she discovered a new molecule that she called actually IDF45, which was a G1 phase inhibitor, but it is now called IGF-BP1. And we all know the importance of this factor. So in the early 80s, she created the Club des Facteurs de Croissance, or Growth Factor Club. And she did that with the instrumental support of the French Cancer Society. So the French Cancer Society supported this meeting, is supporting this meeting since the 80s. So she's been supporting the meeting for 40 years. The club was meeting every year. Early on, it was focused on growth factors, but with time, the scope was broadened. And the meeting covered various cutting-edge issues on different topics related to cancer, and it goes on like that. She died in 2016, and in 2018, the French Cancer Society decided to name this meeting by her name, so it's from Club de Facteurs de Croissance, it became Journée Louise Harel. Thank you for your attention, and then I think we will give the mic or the screen view to Julie Panquin, who is chairing the first session. Thank you, Julie.